In other words, how are you able to generate unheard level of receptor diversity in BCRs and TCRs, which is critical for recognition of epitopes or the antigenic determinants present on different kinds of antigens, right? So this is the topic we are talking about today. And so these are the questions. So we aim to address. Okay. How does antibody diversity arise? That's one. Okay. What causes the difference in amino acid sequence in different antibody molecules or TCRs, right? If the recognition is happening through T cells. How can different heavy chain constant region be associated with the same variable region? Okay, in case of antibody molecule. So this third statement means, so you have a B cell which is making these antibodies initially. So the antibody produced will bind to an antigen X, but the isotype, for example, is going to be IgM. Okay. Later on, the same B cell clone will make an antibody molecule essentially with the same variable region, but with a different constant region. Okay, so that means this is called isotype switching or class switching of antibodies. So these are the things we are going to talk in maybe next 2-3 classes. Okay. First, I will give you an overview how actually things are happening, and then we'll try to discuss in a little more detail about the fine details of these processes. Okay. All right. So this is basically a very simple cartoon showing basically how things are going to happen. Okay. And this summarizes stages in B and T cell development. And the purpose is to ensure that you select your cells of right antigen specificity. And if something is autoreactive, so that somehow is eliminated as these cells are developing, right, in primary lymphoid organs, so that you minimize your chances of getting autoimmune diseases. So that's the purpose. Okay. So you know that lymphocytes, just like any other cells, also start their process of development from hematopoietic cells, right? Hematopoietic stem cells give rise to lymphoid stem cells. And if these lymphoid stem cells have to become B cells, they migrate, rather they remain in bone marrow, because this whole process actually is happening in bone marrow, right? In higher vertebrates like humans, animals, right? So in lower form of animals, so this might occur, for example, in kidney marrows or elsewhere. Okay, the site where these processes take place, so might be different, but essentially the sequence remains same. Okay, lymphoid stem cells are going to give rise to, for example, B cells. They remain in bone marrow. If they are going to give rise to T cells, they migrate from bone marrow to thymus. Okay, and complete the rest of the journey in thymus, right? Let's talk about the B cell development. Okay, once these cells enter in bone marrow, so the first process that has to happen is arrangement of the heavy chain. Okay, and remember, so all these processes actually are happening in nucleus, right? And after rearrangement, so then you get the transcription and translation of the rearranged sequence that you have now in the genome of these cells. right? And these things then are exported onto the surface to facilitate antigen recognition. The first thing that has to happen is rearrangement of immunoglobulin heavy chain gene, right? If these cells fail to do it, okay, so obviously they are going to die, right? So let's say you have generated, you have rearranged immunoglobulin heavy chain, okay, this will not come onto the surface of developing or differentiating B cells. For it to happen, so it has to attach with a surrogate of light chain that we call as, we'll talk about what exactly is the name, but this complex, we do not call it as BCR, we call it as a pre-B cell receptor, okay, because this is not the right light chain that is supposed to be there in the B cell receptor, okay. This is B pre-B or something like that. We'll talk about the details of this process, okay. But surrogate of light chain actually is assembled, and then this complex actually is present on the surface. Now, you have more or less assembled a B cell receptor, although we call it as pre-BCR, but it starts recognizing antigens at this time point. Okay. Now, if this is not able to, or rather, if this is actually recognizing your own antigen, okay, because in bone marrow also you have your lot of these cells keep on making stuff, and you really do not want to select any antibody producing cells which recognize through its product, that means the antibody, your own antigen. So these cells have to die, okay. Or if it fails to recombine this immunoglobulin heavy chain gene, and if it fails to assemble with pre-BCR, all these processes are going to kill significant number of these cells through the process of apoptosis. The cells die, okay. Once you have assembled, and these cells now actually can go to the next stage, okay. Again, next stage is also completed in bone marrow. The cells have to be present in bone marrow. The first thing that has to happen is rearrangement of heavy chain. Now, these cells which have rearranged their immunoglobulin heavy chain gene, they will start assembling immunoglobulin light chain, IGL gene rearrangement. So the first thing is for the heavy chain, second is light chain. Okay, you again generated, now you have this pre-BCR is removed, okay. So this black structure that is shown here is removed with this the other color that actually is also present in the BCR, not exactly the color. So basically now we are talking about the heavy chain and light chain, okay. Now you have a fully assembled B cell receptor, and if it recognizes your own antigens, so the cell is going to die. This is called a second checkpoint, okay. The purpose of having these first and second checkpoint is to eliminate those cells who have either failed to assemble the BCR, or they start recognizing your own antigen. Okay, now let's say if the cell is not recognizing your own antigen, so it will pass through the process, it will be selected, right? And eventually this BCR expressing cell will populate periphery. It will be present in secondary lymphoid organs. Okay, it will circulate. It will be found in the blood. Okay. Now, when you encounter with any antigen, so the cells will start responding to that antigen by recognition of the antigen through BCR. It will then undergo the process of activation, differentiation. It will make activated B cells. These activated B cells are going to produce antibody molecules, right? So this is for the B cells. For T cells, as I said, all these processes of rearrangement are taking place in thymus. Okay. The cells have entered into thymus. They start undergoing, obviously, they have to actually keep dividing. And in the dividing cells, so they have to start this process of rearrangement of pre-TCR. If they fail to rearrange the TCR, right? And initially, just like what you have it for B cells, the initial rearrangement is for the heavy chain and then light chain. For TCR, so the first rearrangement has to happen for TCR beta gene, okay, and then TCR alpha gene. Okay, now there is actually a lot of similarity in developing B cells and T cells as far as they have to actually follow the process of their differentiation in primary lymphoid organs. For B cells, this is in bone marrow, and for T cells, this is occurring in thymus. Okay, you have generated a pre-TCR. Okay, the next stage is, let's say if it has passed through this first checkpoint, so it will start then rearranging TCR alpha gene. Okay, once you have fully formed this receptor, now it will be scanned whether this TCR is recognizing your own antigenic peptides, okay, or it doesn't. If it recognizes your own antigenic peptides, own means the self and non-self recognition and the ensuing events actually are taking place at this time point. Okay, if it starts recognizing your own tissue antigens, and for TCR we call them as epitopes or antigenic determinants, they are essentially this sequence of 8 to 9 amino acid or peptides, right? For cytotoxic T cells, and for CD4 T cells, that means the helper T cells. So this is actually larger, 13 to 14 amino acid sequence. Okay. If they recognize your own antigen at this time point, so the cells die. If they don't, they follow the next stage of their development, which is basically one additional step that you see for T cells, that is actually not present in B cells. What is that? So now you see these two molecules here, right? One of them actually is yellowish and another is purplish, right? Now, one of these molecules actually is CD8 glycoprotein and another is CD4. Now, how is it decided that this cell, whether it's going to become CD4 or it's going to become CD8, right? For a rearranged TCR, okay, if it recognizes peptide which is loaded onto MHC class 1, okay, TCR recognizing peptide loaded on MHC class 1. So this will downregulate CD4 molecule. If the peptide recognized by this TCR is present on MHC class 2, so this will downregulate its CD8 molecule. So essentially in the next stage, so you have either one of these molecules present on the surface. So remember, in thymus, I showed you that there is, when you are actually following T cells in thymus, so you find a lot of double positive cells. And double positive means these cells are expressing both CD4 and CD8 molecule on their surface. Do you remember that? I showed you in one of the fact plots.
Yes, sir.
Yeah. So this is that stage I'm talking about here. So the cell is already having a rearranged TCR. It has both CD4 and CD8. And after this, when basically it has to make a decision whether it becomes CD4 or CD8. So the interaction with MHC class 1 or MHC class 2 is critical. Okay. Obviously, if it is not making any contact with MHC class 1 or class 2, it's going to die, because this is basically a positive selection, right? If it recognizes your own antigen or own peptide coming from your own tissue antigens, this is going to die, okay? Because you have to ensure that the cell is not autoreactive, right? And after that, so for example, if it is reacting to peptide loaded onto MHC class 1, the cell is going to become cytotoxic T cells. The other name of this is called CD8 positive T cells. And in so doing, so it has to downregulate the counter molecules, the CD4. And if it recognizes peptide loaded with MSC class 2, this will retain MSC this CD4 molecule, but it will downregulate CD8. Okay. Now, once you have a rearranged TCR receptor which is not recognizing your own antigen and is expressing either CD4 or CD8, so these cells are now ready to be exported out of thymus, which actually is a primary site for differentiation of these cells. Okay, also considered as primary lymphoid organs, right? Now these cells will migrate to lymph nodes, obviously through circulation. Okay, in lymph nodes, so they are waiting there for recognition of any foreign antigen if you encounter subsequently. Okay, so is it clear how basically how actually you see these processes occurring starting from hematopoietic stem cells to generate either B cells or T cells, and T cells also in CD4 and CD8 T cells. Okay.
So,
yeah.
So why do we see only a small part of the surrogate alpha chain in the pre-TCR instead of a complete one?
Small part means, yeah, well, because the size is not exactly the same. Okay, so it's just rather you call it as a chaperone. So it helps basically exporting the TCR or in case of B cell receptors, so this BCR on the surface of these cells. Because if it is not assembled with either this surrogate chain for TCR or BCR, it will not get onto the surface. Rather, if I might add something here, for BCR, so this surrogate light chain, okay, so we call it a surrogate light chain. Actually, this was discovered by who? Can anybody? And there is a reason why I'm asking that. No idea, right? This was actually discovered by Shiv Pillai, working at Harvard Medical School. Okay, so this surrogate light chain, a very famous immunologist, currently at Mass General Hospital in Boston. Yeah, so there is a one question here. So if differentiation into TH and TC is not successful, well, obviously the cells have to actually undergo apoptosis. For example, if there is some issue here that these cells fail to downregulate one and keep on expressing one, so obviously the ultimate fate is death of these cells, right? So now, for example, if someone tells you that you have to demonstrate the process of apoptosis, okay, cell killing, apart from developing embryo or the model system that you're working with. So these are the sites you should be looking at, okay, the primary lymphoid organ, either bone marrow or thymus, okay. Normally, apoptosis is also happening, for example, in secondary lymphoid organs. For example, if you're infected with something, the cells expand, they die, and eventually you are left with only a pool of these cells. But you really cannot demonstrate cells undergoing apoptosis in these secondary lymphoid organs. And the reason is these apoptotic cells are very efficiently phagocytosed by your innate immune cells. Okay. But if you want to actually demonstrate this process of apoptosis, so thymus is the site. And if you actually want to study apoptosis, so you actually have to work with either thymus, which is easy to isolate, or bone marrow, which is not so easy to isolate. Okay, but these are the two sites you would like to focus your analysis on if you want to study apoptosis. in a fully developed organism, for example, mouse or any other organism that you are working with. Okay, you can actually demonstrate apoptosis happening there. And essentially what that means is, if, for example, if you start with, at this time point, so if you basically make, for example, 1 billion cells, out of 1 billion, only one is selected at this stage. Okay, so one might consider that this is a lot of wastage of energy. Okay, wastage of energy is fine, but you have to make sure that you don't select any cells which are autoreactive. You don't have to select any cells which are not functional. Okay, so all these processes are ensured at the cost of a lot of energy being utilized during the development of your B cells and T cells in primary lymphoid organs. Okay, so so we were talking about basically how you can generate these. Rather, even before that, I think I should show you a different slide, not this one. Maybe this one, because this will put things into perspective. Okay, this one. Okay, so let's talk about this thing. That's your cell started their differentiation process into T cells. Initially, you find them in bone marrow. From bone marrow, these cells migrate to thymus, right? Let's say this is thymus. The first thing that has to happen is the cells keep on dividing, and during the division process, they also start rearranging their VDJ for formation of this TCR beta chain. Obviously pre-TCR, because simply having a TCR beta chain on the surface in these cells, so it's not going to generate any kind of recognition module. At this stage, we call it as pre-BCR. Okay. Cells keep on dividing. The first process that has to happen is this VDJ recombination. And after this thing, again, cells keep on dividing, and all these cells are not going to make to the next stage where they can actually start a rearrangement for the alpha chain, or in case of B cells, for the light chain. So all these cells are going to die through apoptosis, right? And once you have a TCR expressing cells formed, then you start the process of alpha chain rearrangement. Okay, right? Again, so not everything that you have actually generated here, so is going to be selected, because here at this stage you will determine whether the cells are reacting to your own antigen or not. If they react to your own antigen, they are going to die. If they don't recognize your own antigen, they are going to survive. Okay. Just to think, just to basically, in continuation with what we talked about for B cells and T cells, the first thing that has to happen is for the TCR beta chain rearrangement for T cells and for heavy chain in B cells, and followed by this, you have the rearrangement of TCR alpha chain in T cells and light chain in B cells. And during this process, the cells keep on dividing. They keep on making all these kind of processes, and eventually you are left with only very selected few cells which can make to the periphery, and they are actually now sitting there to recognize any antigen that you are encountered with subsequently. All right. So let's now go back to the same thing. So how basically this VDJ and VJ rearrangement actually is taking place. Yeah, so this slide. Okay, so so we call it as generation of diversity. If you want to call it diversity, so feel free to do that. Okay, the process is called recombination of BCR or recombination of TCR. Okay. And what are you recombining? You are actually recombining these different segments that are actually present both in the genomic, in the chromosomal locus, which encode for the heavy chain or the light chain. Okay, so let's say you are following from 5' to 3', the sequence of the chromosome that actually has a gene for your heavy chain. Okay, this is still actually present in differentiating B cells. Okay, these are not fully differentiated cells. So what you see, so you actually starting from 5', 3', you have actually these modules. Okay, so that we represent with V. V stands for variable. D stands for diversity. J stands for joining. Okay, this is called VDJ, VDJ recombination, right? And this is basically for the heavy chain, and for light chain, so you don't have D segment here. Okay, so you have only V and J, right? And then you have also these other, for example, these oval structures. So there are different names for these. So we call them as recombination signals. Okay, or RSS, recombination signal sequences. Okay. Now, the first thing that has to happen is for the heavy chain in your developing B cells. Okay. So what you do is basically, this is actually the very first process. One of the Ds combines with one of the Js. Okay. And this process occurs in a very random fashion. So it can be that you might pick D1, J1. It could be that you pick D1, J2. Okay. D1, J3, and so on and so forth, depending on how many V, how many D, how many J are present. Okay. So through a very random process, so you can select one of the Ds. Okay. It can combine with one of the Js, and once this process is successful, and this is basically nothing but cutting and pasting, right? And when you cut and paste a thing, chances of generating errors are high. So again, if, for example, if this process is not successful, the cell is going to die. A lot of death is going to happen during the recombination process, right? Now, what happens is, if you already have recombined one of the Ds, one of the Js, so all these sequences on that locus of chromosome, okay, what are basically intervening? For example, D3, J1, J2. So these things are looped out. So these are removed. Okay. You can still have D1, J4, or J5, J6, J whatever. All those things are still present in the chromosome, right? But the interspersed sequences are looped out, and these basically have different roles to play during the differentiation process in these cells. But remember, so once you already have formed, for example, one combination, let's say D2 and J3, then you cannot form D2 and J1 in the same cell, okay, on the same locus, okay. And you know, there are two alleles in a cell, okay. It can happen exactly the same way in a different allele, but not in the same allele. Obviously, you cannot utilize, for example, D3, because D3 is looped out on that locus. So all these recombination events you already have blocked by a successful D and J recombination event, right? Once you have achieved this, so then one of the V's out of many combines to complete this process of VDJ recombination. Right. Now, this still is happening at the level of DNA. Okay, so these are basically segment of DNA which are undergoing the process of recombination. Okay, you already have rearranged VDJ for the variable region of your heavy chain, which eventually will be translated and transcribed and translated into this product, which is going to give rise the variable region of the heavy chain. Okay. Similar process also occurs for light chain. Okay, what is missing here is Ds. All these Ds are not present. So that means the diversity that your light chain is able to provide is limited. Okay, but nonetheless, so you have these different V's and different J's. Okay, so here you don't have to follow this DJ thing because D is actually missing. D is not present in the on the locus of this light chain gene on the chromosome, right? So you combine randomly one of the V's with one of the J's right away. And once you have successfully combined this thing, all these segments which are actually interspersed, so they are looped out, this can still have the potential to rearrange. For example, if let's say if this process was not successful, so this cell is going to attempt to arrange one of the segment at the 5' or at the 3'. Okay, and it will try to exhaust all these options in so doing. So essentially what you have done is, so you have recombined one of the V's and one of the J's as a DNA product in your cells. Okay, now if that happens, so obviously this has to undergo the process of transcription and translation in nucleus and eventually in the cytosol to give rise to this variable region of the light chain. Okay, so this is VL and this is VH. Okay, this actually site, a combined basically site constituted by VH and VL is going to give rise to what we call as the paratope, the antigen binding segment. Okay, obviously the other steps also have to take place, that this variable region has to combine with the heavy chain, right, and and also with the light chain concentration. Okay, and those sequences actually are present. For example, you have this VDJ. There is a certain way how these things are organized in the genome, right? And at a distance from here, so you have the gene for the constant region for light for heavy chain, and in this case constant region for the light chain. So this has to combine, and then eventually this combined locus will give rise to the product as BCR. So is this process clear, or you want more clarification on this? Obviously, we are going to talk about this process in more detail, but this is to make it clear in very simplistic form that these are the sequences that are going to happen. For heavy chain, first we have a D and G recombination. Following a successful D and J recombination, then one of the V's combine to complete the process of rearrangement of this variable region in heavy chain, which eventually will give rise to the VH portion. Same thing for light chain, except that you don't have this DJ happening here because D is missing altogether. So there is only one step, which is actually V and J recombination. This will give rise to the variable region of the light chain, and then it will combine with the constant region, and eventually you will make a proper BCR. Now, there are a lot of these V's, lot of these D's, and a lot of these J's in humans, and also, for example, in mice. There are a lot of those are present. And what essentially that means is, depending on even one subtle change at this time point, for example, you have combined D2J3. This will having one antigen specificity. If you combine 3-1, another combination, another specificity. Okay, 1-1, one specificity. 1, 3, 1 specificity. So this process will keep on happening, right? And remember, so once you have a successful recombination event happening in one cell, this cell is not going to actually rearrange the same locus again. Okay. Obviously, all these processes, you don't have only one B cell, right? You have numerous B cells which are actually following this process of recombination, and that is why these different B cells that you develop will have one antigen specificity or the other, or the other, or the other. One clone might recognize one antigen, the others have 1 millionth antigen, one have 1 billionth antigen, and so on and so forth. This is basically a cutting and pasting mechanism. Okay. So this is just to show that you can, depending on what you have recombined, so you are going to generate whatever the number of specificities you can achieve. Okay. Not only this VDJ recombination or VJ recombination provide you the specificity, so the inaccuracies that you have during the cutting and pasting process, right? Recombination, inaccuracy, and then addition of some of the additional nucleotides at N and P, then gene conversion, point mutation, all these things are going to give you more and more and more antigen specificities. Okay. And through all these processes, your B cells, whatever the number of B cells you have, so they will have different receptors, particularly in the variable region. And these different BCRs can recognize one antigen, 100th antigen, 1 billionth antigen, 1 trillionth antigen, and whatever the number of antigen that actually are ever present. So they all be recognized by your differentiated B cells. There is some question here. These VDJ regions must be susceptible to SNPs2 to increase the diversity. Yeah, well, I think this is what you probably want to mean, that you can keep on adding these N nucleotide addition and gene conversion and point mutation and all these processes. Yes, obviously. So, and whatever the new nucleotide you are going to add, so you are going to actually generate a new specificity. More or less, is it clear? I'm pretty sure fine details I have not really talked about, but did you get the idea? So how this process of recombination of your BCR present on B cells actually is taking place?
Yes, sir.
Yeah, okay. Same is the case with your T cells. There is uncanny similarity between TCR beta chain and heavy chain, and TCR alpha chain and light chain, okay, for B cells and T cells. So in BCR, for example, in TCR, so you have again a lot of these V's, lot of these D's, and then J's. However, the organization might be different. Okay, so for example, here you have one of the D's and then several J's. Okay, another B and then several J's. So in some cases, it could be that you have one D, one J, one D, one J, one D, one J. So that actually you have to just remember. So in which species, what is the organization? What is the genomic organization of these different segments of your TCR and BCR? Okay, so this is basically for you to cram. But essentially the processes remain same. So one of the D's combined with one of the J's. Once you have achieved this recombination of D and J, this follows associating with this recombined D and J, okay, one of those V's, and then you generated this VDJ. This VDJ is going to give rise to the TCR beta chain variable region of the T cell receptor, right? And same is the case for the alpha chain. Okay, so very similarly that you observed for the light chain, so you don't have D in alpha. Okay, one of the V's combined with one of the J's, and once this recombined segment is present in your developing T cells, this will give rise to this recombined locus. After transcription and translation process, this will give rise to TCR alpha chain of your T cells. With VDJ recombination, you get the TCR beta chain formed. Okay. Now, so by this process, so basically they estimated what diversity you could generate. So this was found to be about 10 raised to power 15 for T cell. Okay. 10 raised to power 15 would be how much? A lot, right? In humans and mice, so basically this is how it is. This actually is the calculated. The people actually have shown experimentally that this antigen is recognized from this. This TCR is recognizing this peptide, the other peptide, and so on and so forth. This is the experimental data. This is the theoretical data. Obviously, whatever you predict theoretically, so you cannot really demonstrate that each peptides is able to recognize your TCR or BCR, but essentially, so you can actually show a lot of antigen being recognized by your TCR, right? If you want to read more about it, so you can actually go through this paper, Nature Review Immunology, 2006. Okay, so in B cell receptor or differentiating B cells, okay, once a specific BCR is arranged, okay, and is displayed on surface of these B cells come into the periphery. Obviously, you have to make sure that these B cells which express the specific BCR on the surface, they are not autoreactive and so on and so forth, right? So that process ends there. But in case of a TCR, so there is another level of complexity, and that complexity is provided by MHC molecules. Okay. Predominantly, there are two MHC molecules. Okay, so you have MHC class 1, MHC class 2. MHC class 1 can recognize shorter sequence, and this actually decided by the structure of MHC class 1 heavy chain, right? And then you have the beta 2 microglobulin associated with it. The size constraint for MHC class 1 molecule for the peptide recognition are provided by heavy chain of your MHC class 1 molecule. So that is why this is able to accommodate shorter sequences. But for MHC class 1, the size constraint is not there, so that's why it can recognize longer peptides as well. Up to 13, 14 amino acid long peptides can be easily accommodated by MHC class 2. And obviously, this is going to activate your CD4 cells. This is going to activate your CD8 cells. Now, one thing that you have to remember at this time point, although I will revisit this issue, that not every single amino acid that is present in the peptide interacts with MHC molecule or with T cell receptor on the other side, right? So there are only certain positions which actually make contact with MHC class 1 or MHC class 2. Okay, the amino acid which actually interact with MHC molecule are called anchor residues. They are very actually specific with some exceptions, but more or less at P2 position or P9 position, so you will find anchor residues. And P4 or 5 position, so you find TCR contacts. So these amino acids are going to interact with TCR, which actually we already have talked about. You have TCR alpha and beta chain, and then you have accessory molecules like CD3. Then you have the other molecules like these gamma and epsilon and zeta chain for signal transduction processes. But the recognition of peptides actually is through the alpha and beta chain variable region. This is that variable region. It recognizes one of those peptides, one of those amino acids present in your peptide, right? So so basically, yeah. The point I wanted to make in this slide was that you have provided your T cells the ability to recognize different peptides, which are loaded onto MSC class 1 or MSC class 2, depending on whether you have CD4 or CD8 T cells, right? Now, MSC molecule is one of the most polymorphic molecules in your system. So essentially what that means, these MSC molecules can load on their surface, on basically on the pocket, which is in this pocket where these peptides actually sit in. So these peptides, what will be loaded on MSC class 1 will be decided what MSC molecule you are talking about. And there is huge polymorphism in MSC molecules, which actually provide further diversity in antigen recognition. Okay. So essentially what that means is T cells can recognize more number of antigen or antigenic peptides than B cells. Okay, so how are the antigen processed in the such cells that they have such specific anchor residues to be loaded onto MSC class 1? We'll talk about it, don't worry. So when we talk about antigen processing and presentation events, we'll talk about how these processes are taking place. So you have a lot of these peptidases. For example, you have this proteasomal complex. You have these different peptidases which are actually present in lysosome or endolysosome. They actually recognize certain residues and cut it there. Okay. So this is not to say that you only generate 9 mers or 8 mers or 7 mers. Okay. Obviously, with this, there are a lot of these peptidases which can keep on cutting at all these species, and you can generate single amino acid, 2 amino acid, 3, 4, 5, whatever. But when you achieve a specific sequence, that can be loaded on MSC class 1 or MSC class 2. Okay, so yeah, so we already have talked about this slide where your cells were coming from bone marrow. They enter in thymus, they keep on dividing. The first process that has to happen is VDJ recombination followed by VJ recombination. Then you generate complete TCR, and then this is basically probed against your own antigen recognition, right? If you find autoreactive cells, the cells die. If they don't, they survive and they get to periphery. Yeah. So again, if you summarize these processes, basically the developmental processes of T cells for a particular specificity and basically to impress upon you that you start with so many cells, but you end up getting very few cells in periphery. Okay. So the first process is the positive selection, then the negative selection, and then these cells come to periphery, which have been actually successfully passed through the process of positive and negative selection. Positive selection means, for example, if the rearranged TCR is not able to recognize anything, if it does not recognize any MSC molecule, okay, if it is not recognize any of the MSC molecule that your different immune cells or different epithelial cells or all the kind of cells that you have in thymus, if this MSC interaction is not happening through TCR, obviously through outside the peptide binding groove, so this process is called positive selection. And what that means is, if you have no interaction, cells die. If you have interaction, cells survive. Negative selection is where the cells are sensed for autoreactivity. Autoreactive cells die, foreign antigen reactive cells survive, right? And then in periphery, so you come up with these cells, which are largely reactive to foreign antigens and very few cells which actually are shown here. Okay, they might be potentially autoreactive. Okay, maybe let's say as because this process is happening through all these processes, right? All these steps. So you might select some of the autoreactive T cells, and which actually are potentially damaging. In order to control the activity of these, although you have made sure that not many cells actually pass through the process of positive and negative selection and be present in periphery, but some autoreactive cells come in there, but their activity is blocked by the regulatory T cells that you have. Okay, and it was shown experimentally that if you somehow deplete these regulatory T cells from a model organism, for example, in mouse, so these animals, they start developing all kind of inflammatory diseases. Okay, in mouse, we call it as scurfy disease, and in humans, so this is called as IPEX syndrome. Although, good thing is that these are very rare events where there is mutation in FOXP3 locus. Okay, so you actually have a very good system to avoid any autoimmune diseases. Okay. Yeah, so we are going to now talk in little more detail about these processes of recombining BDJ, and then how it was actually shown experimentally, and basically then how this variable region actually is able to combine with the constant region, and having the same variable region and different constant region. So all these processes we'll talk about in subsequent slides. If we have time. No, we don't have it's time. It's 9:45 already, 10:45 already. But if you have a quick question or so we'll take it. Otherwise, we'll continue in the next class.
Sir?
Yeah.
Are there communications between B cells and T cells to make sure they don't generate the same kind of combinations to reduce redundancy?
Well, remember, your B cells, through their BCR, recognize the whole antigen. Okay, I have not really talked about that, but briefly I did mention that. So the antigen does not have to be processed for recognition via BCR. So you have injected, for example, spike protein directly, or a receptor binding domain, or HA protein of influenza, so that can directly is able to stimulate your B cells. But for your T cells to be stimulated, so this protein has to be internalized. It has to be cut into those small pieces, and those snippets, which are derived from this antigen, this big antigen. Okay, and that we talked about 7 amino acid, 13 amino acid, 9 amino acids, 8 amino acids. Those are loaded onto appropriate MSC class 1 or MSC class 2. Okay, and then your T cells can recognize it. So the mechanism for B cell recognition and T cell recognition of antigen or antigen derivatives is different. Obviously, chances are that some of the epitopes that you generate, for example, which can recognize your TCR present on, particularly on CD4 T cells, can also activate your B cells. There exists a possibility, but these are more or less exclusive events, right?
So more specifically, I was, I guess, asking, let's say a B cell makes a recombination with a V1D2J1, and another B cell also makes the same recombination.
Well, that can also happen, and but that event actually is very rare. Although, so there is no crosstalk between two B cells. Okay.
Correct.
Although at this time point, rather, I would say these processes are not really well understood. Highly unlikely the rearrangement has to happen in a very random fashion. So two B cells can still recognize the same antigen. And if you actually calculate experimentally, or people actually have shown, approximately 500 to 1000 B cells can actually express the same antigen, same receptor, same BCR. Okay, for one particular epitope, so you can have cells, B cells, which actually are from 500 to 1000. So it's not like for one epitope you'll have only one cell, and that actually is a good thing, because let's say if something wrong happens to this one particular T cell, so that means you actually lost that specificity, right? But it's not like that some of these specific sequences are preferred and some are not. This actually is a very random process, and that is why you are able to actually generate all kind of specificities. I think someone else is logging in for the other class, so we have to actually stop this here.